we get experimental science. We're curious, non-judgmental.
All right, Andrea, this is the piece with the cordyceps in it. We got our little ant in here. This is 30 million years old. But before we start looking at the sample, let's learn a little bit about cordyceps. So the piece is huge, Andrea, but if you look at the ant, see there's the ant. It's a regular carpenter ant size. It's just caught in a very big piece of resin. So Andrea saw one of these clickbaity videos, and she was asking, she's like, well, what about this is true? What about this is not true? So that's what we're gonna touch on. There are cordyceps samples that are even older than that. So the idea is most insects actually have a cordyceps that infect them. So a cordyceps fungus that is specific for an ant, one ant species. So there's one for bullet ants, there's one for carpenter ants, there's one for leafcutter ants, and then there's others for other kinds of insects that they just are very specialized on. When the animal first gets infected by the cordyceps, it's just a spore that goes through their exoskeleton. And then that spore grows in their body and goes to their brain. First behavioral change that they have that lets you know that maybe there's some kind of infection is they start overly cleaning themselves. And it's just this excessive cleaning that you can't really explain, and just not something that they tend to do. So Andrea, where it gets especially wild is that these animals are not aware of what they do. So this animal, once infected, climbs up a blade of grass, bites into the blade of grass, and dies. It is not doing that voluntarily. It is not like, hey, you know what would be really cool is if I went and died on the tip of a blade of grass. It does not think that. But the reason I'm highlighting it as like, Balint, then why are you talking about it? That's kind of weird. Well, I think their behavior is super weird. This is the fungus that's highlighted in the TV show and video game The Last of Us. So there is no chance that in its current form it's going to jump species. What the fungus relies on is how the insects breathe to get its oxygen. So an insect breathes, there's no lungs, there's no circulatory system in terms of veins and arteries. Rather, there's just free-flowing hemolymph throughout the body of the animal. And they breathe through these little tubules that come from the exoskeleton inward, and then oxygen diffusion takes place. And the fungus needs that kind of oxygen in order to survive. It will not be able to just get into a bloodstream where there's not immediate access to oxygen. And so it's not going to be able to take over a human, unlike the TV show or the video game. The TV show and the video game Last of Us, it's a really cool idea. It's the science-wise of what it does to the insects is correct. It's just the translation to mammals just can't happen at this stage. The ants can detect that these animals are infected, and there's even a trash pile for the dead or dying away from the nest site. So this ant has been infected in its brain. It climbed up a blade of grass, and from its head is erupting a spore. So it's a 3-week time lapse, and then the spores go and then all neighboring ants also get infected as well. So, Andrea, if this successfully does what it needs to do, these ants will erupt the spores over the colony and just destroy the entire colony. The fungus actually drives them to go a particular height. And so it's not fully understood what exactly happens. Right now, there was a paper that came out maybe 2 years ago, and what they tried to do was they did sequencing to see what genes are differentially expressed in the brain of the animal upon infection. There was a lot of difficulty in this because it turns out in the lab, the infection method that they have is only good about 50% of the time. And the first 50%, the animals don't get infected. Of that remaining 50%, 30% of those actually go to completion in terms of this happening, right? Otherwise, there's usually either they're not infected at a sufficient enough dose and they fight off the infection, or they don't actually know if it's the infection is being fighting off or not. We just don't know. But it seems like the fungus gets the ants to go on a particular substrate, climb up on a particular branch of sorts, and they go to a particular height. So the fungus seems to somehow drive the animal to go to a particular height before this next event takes place. And so there's a lot of detection of hijacking in the brain that happens on these animals. The length between the stalk of the fungus and the death of the animal. So Saphira's mama, at this stage, it's dead. So the last thing the animal does is it grabs onto that blade of grass and dies. And then the fungus starts coming from its brain and head. But from infection until that bite, see, that's the difficulty. It's doing it in a lab setting where you're controlling for timing. That's what I was saying, it's not a good enough infection strategy where it gets infected 100% of the time. And that you can actually get those good measurements in there. But I believe it's on the order of a week that that infection was taking place up until like the death of the animal. So for the sequencing, they weren't able to identify in a good, clean way different genes between the different organisms. So like from what were ant genes, what were fungal genes, and how they interacted were difficult to parse out. So all they got was a list, and they didn't go to the next steps. Like Andrea, the next step would have been, right, if you identify the genes from the Cordyceps fungus, and, you know, you hypothesize, okay, maybe there are these genes that get turned on in the Cordyceps, and then in the brain of the animal, different genes get turned on or off in a different manner, right? It would be cool if then you artificially, if you, without infecting the animal with the fungus, change the gene expression in the animal's brain. Can you make this ant climb up the blade of grass and bite into the grass and die without infecting it with the fungus? Like, can you do it with just genetic manipulation? Andrea, they look very different. So there's different types of cordyceps for different insects. The reason it can infect mammals is because the way these animals breathe, they breathe through their skin. They don't have lungs. If they had lungs, then it would be a very different question. But because they have this open circulatory system, meaning that there's no veins or arteries, there's no lungs, there's just these little tubes that come in from the skin, and they diffuse the oxygen throughout the body. That's where the fungus can permeate and get its oxygen that it needs to breathe and survive. And so we don't function that way, so that's why they can't survive in us. Heat might have something to do with it, but definitely the way that it would get oxygen for it. So this is our 30 million-year-old amber with an ant in it, and right there is the beginning stage of that cordyceps growth. That white mass coming from the exoskeleton of the animal. That right there, that's the scary business. And that's the beginning stage of that fungus breaking through the exoskeleton. Terrifying, right? Isn't it cool, Ian? This is one of my favorite samples. As far as I'm aware, every insect has a cordyceps that can infect it. Why is it a good thing? It's a good thing because it can keep populations under control in terms of like insects, where if there weren't these things to keep the animals like population at bay, then they would just be like overpopulating all areas. So in that sense, it's a good form of population control, as terrifying as it is.